Today I'm going to show our recent results on experimental results on the study of the spatiotemporal propagation of sensory evoked activity in different level of anesthesia. So the project framework here is the Human Brain Project, and in particular we collaborate with many groups in the Human Brain Project with the common aim to develop multi-scale models of brain responsiveness. And in this framework, this project provides mesoscale calcium imaging data and analysis. Before going to the results, I have a couple of slides to recall some important concepts about the processing of sensory information at cortical level. Here I reported 3 examples of the biphasic dynamic of the somosensory stimulation. And as you can see, the somosensory stimulation leads to an early response that is faster and a secondary response or late response that is slower. And the important thing here is that this is observed both at single cell level, like in this group from Peterson, but also from an entire cortical areas. So in this beautiful paper from the Peterson group, they try to account some role to these 2 phases, and they observed that by optogenetically inhibiting selectively the first phase and the secondary phase, they suggest, the results suggest that the Haley component or primary response account for the representation of the stimulus at cortical level, while the secondary response is more related to the subject perception of the stimulus. And without going too much in details, the second aspect that I want to stress here is the spatial distribution and the importance of the broadcasting of the information between different areas. Indeed, in this paper from the Karel Svoboda group, they optogenetically inhibited several areas in the mouse cortex. These mice are trained to perform leaking behavior triggered by sensory stimulation. And they observed that not only sensory areas account for the successful behavior that is leaking behavior, but also other areas are necessary to perform correctly the behavior. So sensory information to be correctly perceived needs to be broadcast and integrated up over different areas, cortical areas. And the last example is this paper from Rosenthal and collaborators, in which they observed that the propagation of sensory evoked stimulus, the representation in the cortex, the direction of this propagation is state dependent. And they observed that in the wake state and under ketamine anesthesia, the direction of the propagation among the cortical areas is different. So summarizing, there are temporal and spatial features that are brain state dependent for the processing of the sensory information at cortical level. So there are many open questions, and we focused on the brain state dependence of the distributed response in 2 different states of anesthesia. And we wonder how the correlation structure changes during the stimulus and the role of the secondary response on the entire cortex. So this is the aim of this project, and to achieve this aim, that is the study of the spatiotemporal propagation of the sensory evoked activity, we used wide-field optical imaging that allows to monitor at the mesoscale level the population of neuronal activity, the transgenic mice expressing GCAMP6F indicator, and isoflurane anesthesia to precisely move within the B-stability regime from deep to an intermediate state of anesthesia. So very briefly, the animal model that we use is the Taiwan GCAMP6F mice, that is a transgenic mouse line expressing the GCAMP indicator only in excitatory neurons. And the GCAMP is a genetically encoded calcium indicator that consists in a protein complex that changes the emitted fluorescence. Is a fluorescence indicator that changes the emitted fluorescence according to the intracellular calcium concentration. So neuronal activity leads to a rapid increase of the intracellular calcium concentration that is reported by this indicator as a change in the emitted fluorescence. With these tools, we studied neuronal population activation at mesoscale using the optical setup that is here schematized, consisting in a fluorescent path with excitation line and emission line that report the signal to the camera, and the reflectance path that used to monitor the hemodynamic in order to subtract the hemodynamic component to the fluorescent signal. And the somatosensory stimulation was achieved by delivering an air path, and here is reported our square centimeter field of view that covers both the cortical hemisphere of the mouse. This technical combination has positive aspect like the high spatial resolution and the specificity of the neuronal population studied, that in this case is only excitatory neurons. While the disadvantage here is that imaging camera needs time to integrate emitted photon that leads to relatively slower temporal resolution. But in this case, in this project, the event that we want to study range over hundreds of milliseconds. So we set the setup to have a sampling frequencies of 40 Hertz that is more than sufficient to study the phenomena. And now moving to the results, first of all, we characterize these two states of anesthesia, controlling the isoflurane doses. And as you can see from the movies, during intermediate state of anesthesia, we saw a significant increase of the frequency of the upstate, and accordingly, the upstate duration also is increased, and the downstate duration decreased. So once characterized these two states, here I reported what happened when we stimulate with 60 millisecondary path, what happened at the cortical level, and is more easy to see this effect with the image sequences. And as you can see, the sensory stimulation elicit a local response in the contralateral hemisphere, let's say the stimulated hemisphere, that is followed by a global wave. By averaging several trials on different mouse and registering the mouse on the Allen Brain Atlas, we obtained this average sequence of activation for both the deep anesthesia level and the intermediate anesthesia level. And the first aspect that we noticed is that for both the levels, the activation starts asymmetrically and become symmetrically. But even by high, you can see that there are differences in the temporal dynamic and also in the level of activation of the different area engaged by the stimulus. And these differences become clearer by performing the difference between these two maps. And here I reported in red the prominent activation of the intermediate anesthesia, and in blue the prominent activation in deep anesthesia. And at a glance, we can say that the intermediate anesthesia leads the stimulus to start, the response to the stimulus to start later and last longer. Then we perform the correlation structure analysis. We perform the functional connectivity matrices on several cortical areas, and over the matrix, we perform hierarchical clustering analysis. And as you can see, as expected, the two levels of anesthesia are mirrored by different structure of the networks. In particular, overall, the deep anesthesia show more heterogeneous connections and less level of correlations compared with the intermediate state of anesthesia. The interesting point is here is that during the stimulation, these two matrices become more similar. It's not completely similar, of course. There are, although some very important difference, but the overall structure become more similar. Probably these results related to the stimulation of the same underneath connection, because it's the same stimulus that we deliver to in both the cases. So here I want to stress an important aspect, a technical aspect. Indeed, with our methods, we can resolve a single trial simulation with sufficient signal-to-noise ratio. Indeed, I reported here an image sequence recorded for a single simulation, and here there is the time series extracted from this region of interest. And as you can see, the biphasic response is still there, while averaging several trials in the same animal leads to almost missing all the feature. So this is the reason of this become clear by looking at the single trials. And as you can see, the secondary response here is time varying. So it's averaged out, let's say. When you average several stimulus, you miss this information. So we focus on the trial-by-trial data analysis. And this is possible with our technique, and it's harder with VSD or other kind of imaging technique. So here I reported the analysis of the time series extracted from some cortical areas for the sake of space and time. I choose 6 areas, both the motor cortices, the somatosensory cortices, the V1 and retosplenial that are in the caudal part of the hemisphere. And on the x-axis, there is the cortical areas, and in the blue background and red background, the right hemisphere and left hemisphere, respectively. This is the delay analysis. The delay is the time from the stimulus to the threshold that is set to time the standard deviation. And as you can see, there is not clear brain dependence of the delay value. So in deep anesthesia, intermediate anesthesia are more or less the same delay values, although we can see a trend in reduction of the delay in the stimulated hemisphere. Conversely, the response duration, that is the time from the first cross of the threshold to the return to the threshold, is significant increase during intermediate anesthesia. And these results could suggest that during intermediate anesthesia, it starts to be more integrated, the signal, the sensory evoked signal. And looking at the primary response, we observe that there is no difference between the deep anesthesia and intermediate anesthesia concerning the amplitude of the signals. Although there are significant differences between the amplitude at single areas, and this is mirrored in both hemispheres, there is not difference between the true brain state. On the other hand, there is a significant difference in the dynamic of the primary response. Indeed, the rising slope is decreased, and accordingly, the time to peak is increased during intermediate anesthesia. And probably, we explain this, we imagine that this is due because when a stimulus impinges on a network with more background activity, probably needs more time to reach its full representation in the cortex. And looking at the secondary response, we have here probably the more important results that we obtained, because the secondary response is dramatically more probable to be elicited during intermediate anesthesia. So this suggests that although in the B-stability regime, we have more probability to elicit a secondary response in a lighter level of anesthesia. So conversely, the overall dynamic of the secondary response is not significantly different between the states. There are some trends and some statistical significances, but overall, there are no remarkable brain state dependence of the secondary response dynamics. So we wonder if this is because the secondary response could be more similar to a spontaneous upstate. We want to perform some next step analysis to understand the similarity between this secondary response and the spontaneous upstate. So these are the conclusions, and the overall take-home message is that our results suggest that within the biosuppression regime, the intermediate level of anesthesia could be associated with an increased degree of sensory information integration. So in the last slides, I want to mention our collaboration here with Kirsten Zhao and Julia. Within this collaboration, we developed an analysis pipeline to extract quantitative information from our imaging data. And here I reported the collaboration to develop an infrared mouse model of traveling waves. And with the ENFN, EDBAPS, and ULIC, we developed an universal pipeline that allows to compare and analyze very different datasets, as in this example, the ECHO dataset from EDBAPS and the calcium imaging from our lab. And last but not least, the collaboration with Nuria and Alan, in which we, the aim is to develop a calcium imaging mean field model that simulated changes in the anesthesia level by modulating the adaptation level. Finally, I want to thank all my colleagues in LENS, ENFN, CNRs, and ULIC, in particular Letizia, Elena, and Francesco Pavone, all the founding agencies, and you all for the attention.